Hello, everybody, to the March edition of the MycoTalks. My name is Iliyan Iliev, and I'm at Weill Cornell Medical College in New York. My co-chair today is Joe Heitman from Duke University, and we have two exciting talks for you. The first one is from Dr. Scott Filler, and the second from Dr. Tihana Bicanic. For those of you who are new to the MycoTalks, those seminars were organized almost now a year ago, and they were spearheaded by the organizing community here on the bottom, and they aired last summer. So the purpose of those talks is to provide diverse science and discussion in the area of medical mycology with the attention of diversity in gender, in career stage, geography, field of science. So in the addition to the all great purposes of the MycoTalks, this is actually also a way for our international mycology community to keep in touch, stay connected during those challenging times. So even today, when we overlap with a landmark meeting in the field, we see that a lot of people are actually in the online room. So we really appreciate that. We appreciate you. So the structure is approximately 30 minutes per talk, and each talk goes one after the other. After the talks, we'll have a Q&A session. So please submit your questions to the Q&A area, Joe and I will actually combine those questions and submit them to the speakers. If you have to leave earlier, or if you want to share those talks, or if you want to just receive them, please go to the MRC YouTube channel here on the bottom. So all that organization actually takes a lot. So I really want to thank the people on the background here from MRC that are helping with this huge amount of coordination. I would also like to remind you that those MycoTalks will go, and here is the schedule for April and May. So please register, that's very important. Also, yeah, I want to highlight this fantastic initiative here by Liz Ballo and colleagues. So those are excellent training seminars, again, in the field of medical mycology. You know, the purpose here is to actually engage students, postdocs, clinical trainees, all the trainees basically, to share their research, and we encourage you to register for those as well. So with this, I will actually move on, and I will present our first speaker. I'm thrilled to introduce Dr. Scott Filler. Scott is a professor of medicine in the David Geffen School of Medicine and chief of division of infectious diseases at Harvard, UCLA. He has multiple seminal contributions to the field of fungal pathogenesis. So here I'm saying on purpose fungal pathogenesis because his contributions, that's a very broad term, right? But his broad contribution is going from diseases that are actually very, very different. So disseminated candidiasis, invasive aspergillosis, oropharyngeal candidiasis, among a few to mention here. So Scott and his team identified ALS3 and SSA1 invasives of Candida albicans and discovered several host receptors of those invasives, which actually led to a number of seminal papers that I hope all of us have actually read. On the Aspergillus fumigatus side, Scott's group and colleagues identified the invasion CAL-A and determine its integrin target. So today's talk, Scott will focus on Aspergillus fumigatus and a transcription factor that regulate metabolite production and virulence. I'm very excited to that, and I will give the stage to Scott. Scott, thank you.
Thank you, Iliyan, for such a nice introduction. It's a great pleasure to be able to to present at this MycoTalks. I think it's an incredible, it's very nice for this talk to be given, and I think the MycoTalks are a great resource for the mycology community. So today I'd like to present some of our work on Aspergillus fumigatus transcription factor expression and function during invasion of the mammalian lung. So the Aspergillus fumigatus is a ubiquitous fungus. It's the most common fungus to be found in compost piles. And in fact, the fungus makes numerous conidia, and we probably inhale between 100 to several thousand conidia every day. These conidia are quite small, only about 2 microns in size, so that when they're inhaled, they can reach the terminal airways and even be deposited in the alveoli. And in a normal host, of course, these conidia are cleared and don't cause any disease. But when patients are immunocompromised, such as by steroids or neutropenia, for example, related to cancer chemotherapy, the organisms can germinate and then invade into the deep tissues. And so these proliferating fungi in the tissues can cause direct damage. And part of this damage is also due to mycotoxins or secondary metabolites that are made by the organism. So although Aspergillus is a relatively common cause of disease, it's estimated there's probably 10,000 cases of invasive Aspergillus in the US every year. It's been relatively poorly studied. For example, in the Aspergillus genome, there are about 10,000 genes, and less than 5% of them have been characterized. So one way to identify genes that are involved in pathogenesis is by in vivo transcriptional profiling. So this will enable us to determine what genes are expressed during infection. And we can learn multiple things from this expression data. For example, we can identify key response pathways and hopefully novel virulence genes. And ultimately, these studies can lead to new diagnostic or therapeutic targets. So I'm going to present some of our work where we've done in vivo transcription profiling with Aspergillus fumigatus. And let me, of course, mention that all of this work was performed in collaboration with Aaron Mitchell and Vinnie Bruno. So here's the challenge of in vivo transcriptional profiling. The way this is done is normally one would infect a mouse with Aspergillus and at various time points harvest the lung and extract the total RNA. Well, when doing so, the vast majority of the RNA that's extracted is mouse RNA, and probably less than 1% of the RNA that you get is fungal RNA. So this makes it very difficult to specifically assay fungal gene expression, especially for genes that have relatively low expression because they account for such a low percentage of the total RNA. Well, one way to get around this is to use nanostring technology. And so this is how it works. If this is an Aspergillus RNA, there's 2 different probes that must hybridize to the RNA. One is the capture probe, and then the second probe is the detector probe. And then we can assay for the presence of this mRNA by this barcoded fluorescent reporter tag. And so this allows direct measurement of RNA. So you don't have to make cDNA or introduce artifacts due to that. It's highly sensitive. And because 2 different probes must bind to the mRNA, it's also highly specific. As I'll show you, it's also reproducible and relatively affordable. So it's possible to assay multiple samples with 1 probe set. So Aaron Mitchell and I helped develop 2 different nanostring probe sets. One was a set of environmentally responsive genes. And these were 97 genes that were culled from the literature. And they were genes that were known to respond to different changes in the environment, such as nutrient limitation, oxidant stress, and so forth. Then we looked through the Aspergillus genome and identified 400 transcription factor genes, virtually every transcription factor gene in the genome. Now, there was some overlap between these 2 probe sets, so that we ended up assaying the expression of 479 genes in total. So because we could test so many conditions, we used the probe set on multiple conditions. So the first was to look at the gene expression in the lungs of immunosuppressed mice. So to do so, we exposed mice to cortisone acetate, high dose, and then put them in an inhalational chamber that delivered an aerosol of wild type Aspergillus fumigatus canidia. So we know that the mice inhale roughly 2,000 to 3,000 canidia per lung. So they're infected by a relatively low inoculum. Then at days 2, 4, and 5, we harvested the RNA from the lungs for nanostring analysis. So in parallel, we also grew the organisms in Aspergillus minimal media, either alone or medium that was modified to have either low iron, low zinc, low nitrogen. And we did this alone and in combination. So for example, some of the media had low zinc and low iron, and we even tested low iron, low zinc, and low nitrogen. We tried supplementing the medium with serum and also grew the organisms under hypoxic conditions. And so, as I mentioned, our data quality were quite good. And let me show you an example of this. Shown on the x-axis is the probe counts from one condition, and on the y-axis, the probe counts from another condition. So here's the probe counts for one RNA, and you can see it's a nice 45-degree angle. And here's the probe counts for a second RNA, again, 45-degree angle, and they're very similar. We get a little variability down here for the genes that have very low mRNA counts, but especially for highly expressed genes, the results are really reproducible. And the yellow bars show the results of mice that were uninfected, and you can see the probe counts are very, very low. So there's a very high signal-to-noise ratio. So Aaron Mitchell took our nanostring data and compared it against about 74 different data sets for Aspergillus gene expression that were in the literature. So shown here in the middle is our data from the infected lungs. So the one thing that our data clustered with was data that was generated by Elaine Bignell. And so she had performed microarray analysis of Aspergillus fumigatus germlings that had been recovered from the lungs of mice by bronchoalveolar lavage. So because she recovered these organisms by BAL, this profiled only early infection prior to tissue invasion. And you can see while the gene expression profiles were similar, they clearly were not the same. Notice this area here, for example. And shown on the right is the gene expression of Aspergillus grown in our Aspergillus minimal media that was depleted of either zinc, nitrogen, or iron. And again, the results look fairly similar to the in vivo situation. So let me zoom in on that cluster analysis and show you some conclusions we can draw from it. So the first is the gene expression in the lungs during invasive growth at days 4 or 5 were actually quite similar, suggesting that Aspergillus fumigatus is roughly at steady state at this time point. Note also that there was clustering again with minimal media that was depleted of either zinc, nitrogen, or iron. And so this indicates that Aspergillus fumigatus experiences iron, zinc, and nitrogen limitation in the lung. This fits well with what's been known because mutants of Aspergillus that have defects in iron uptake or zinc uptake or amino acid utilization have all been found to have reduced virulence in the mouse model of Aspergillus fumigatus. And down here is a cluster with Elaine Bignell's data, the organisms recovered by BAL. And so although they are somewhat similar to what we've seen in invasive growth at days 4 and 5, there's also some distinct differences, suggesting that invasive growth by day 4 or 5 is distinct from the initiation of infection. So we also found that a striking finding was that invasive growths upregulated the expression of secondary metabolite genes. And so shown here is a full change of gene expression relative to growth in Aspergillus minimal media. And these genes here all encode various enzymes involved in the biosynthesis of gliotoxin. And you can see they were massively upregulated in vivo. For example, GlyG was upregulated by over 9,000-fold in vivo. NSCR, which encodes for another mycotoxin, was also highly upregulated in vivo, as was ASP-HS, which encodes a hemolysin. So one thing that was sort of interesting when we looked at fold growth is that some genes that were known to be very important for the virulence of Aspergillus fumigatus were actually not particularly upregulated in vivo. For example, SRBA, which Rob Kramer's group has found is a master regulator of hypoxic growth, was only upregulated by 1.8 to 1.7 fold in vivo. HAC-A, which David Askew's group has shown to be required for or govern the unfolded protein response, was also actually downregulated in vivo. And PAC-C, which Elaine Bignell's group has shown governs the response of Aspergillus to alkaline pH, was also only modestly upregulated. However, fold change doesn't tell the whole story, because here are the normalized probe counts. And so the mean in our 479 probe set, the mean probe count was about 2,200. And you can see SRBA was massively expressed in vivo with probe counts of 16,000. HAC-A was also highly expressed, as was PAC-C. And in fact, these three genes were among the top 30 highly expressed genes in vivo. So it suggests then that many stress response genes are highly expressed in vivo, and they may not necessarily be upregulated because they're already so highly expressed. So we looked through our gene expression data to look for transcription factors that we could study. So we had three simple criteria. One was that they had not been studied previously. The next criterion was they were highly upregulated in vivo, and these yellow boxes indicate those. As you can see, for example, this gene up here was upregulated by 43-fold in vivo, and NSCR was upregulated by 308-fold in vivo. And we also took some transcription factor genes that had very high levels of expression in vivo, even if they weren't necessarily upregulated. For example, you can see these genes had between 11 and 27,000 probe counts, which was extremely high. So Hong Liu in my lab constructed deletion mutants for each of these genes. Then we tested their virulence in mice. And so to do so, we actually used nanostrain. And so we basically infected the mice with either the wild type strain AF293 or each of these mutants, and then extracted their, sacrificed the mice at day 5, extracted their lung, harvested the RNA, and used nanostring analysis. So we had a probe for a ribosomal RNA gene in Aspergillus, and then we looked at the ratio of that expression versus some housekeeping genes in the mouse lung. And these are the normalized expression data. And so one thing that pops out as this is the wild type strain here, is that mice infected with this strain, the RLMA mutant, had significantly lower fungal burden than any of the mice infected with any of the other strains. Also, though, Hong Liu astutely noted that mice infected with this mutant, SLIT-A, also, although they had pretty much wild type fungal burden, the mice just looked healthier and their lungs looked better. And in fact, we looked at the weights of these lungs. You can see that mice infected with the SLIT-A mutant had lungs that weighed less than mice infected with the wild type strain. And so this suggests there was probably less tissue damage and less edema, again suggesting that the SLIT-A mutant might actually have a defect in pathogenicity. So to verify this, we complemented the RLMA and SLIT-A mutants and tested their virulence in mice again, but used survival as the endpoint. As you can see, mice infected with the RLMA mutant, shown in red, lived significantly longer than mice infected with either the wild type strain or the RLMA complemented strain. However, shortly after we performed this experiment, a group from Brazil, led by Gustavo Goldman, published this paper here, showing that RLMA is not only required for virulence, but it governs cell wall integrity through this pathway as shown here. So we next turned our attention to SLIT-A. So again, we found out the SLIT-A mutant was less virulent in the mice because mice infected with the mutant lived longer relative to mice infected with the wild type strain or the SLIT-A complemented strain. So to try to determine how SLIT-A governed virulence, we first looked at some in vitro data. So this is some stress response data. And we found here that the SLIT-A mutant was defective in growing on protamine and SDS, both of which are cell membrane stressors. However, it had wild type growth on caspofungin, Congo red, and chalcofluorite, suggesting that it did not have any defects in cell wall integrity. And it also grew normally in the presence of hydrogen peroxide indicating that it did not have a defect in oxidant stress response. Now, just a month or so ago, a group from Nanjing published that a SLIT-A mutant has defects in ergosterol synthesis and is hypersensitive to azole antifungals. And so this defect in ergosterol, which is a key membrane sterol, may actually explain why it's hypersensitive to protamine in SDFs. So we also looked at the interactions of our SLIT-A mutant with host cells. And so we first measured the capacity of the organism to invade the A549 type 2 alveolar epithelial cell line. As you can see, the organism was quite defective relative to the wild type strain or the complemented strain. It also had reduced capacity to damage epithelial cells. However, it had wild type susceptibility to being killed by mouse macrophages. So to get a real view of how SLIT-A governs virulence, we performed RNA-seq. And so we knew from our nanostring data that growing organisms in the presence of low zinc and low nitrogen induced a transcriptional response that was pretty similar to what was seen when the organism was grown in vivo. And so we did RNA-seq for the SLIT-A mutant. And so what we found is many different secondary metabolite genes were just had altered regulation in this mutant. So to give you a little background, Aspergillus, the Aspergillus genome contains roughly 33 secondary metabolite gene clusters. And it's known that in Aspergillus, in fact, many filamentous fungi, the genes for a given secondary metabolite are arranged in clusters on the chromosome. So for example, this is the gliotoxin gene cluster. So each of these genes here encodes an enzyme in the gliotoxin biosynthesis pathway. And also another factor of these clusters is they almost always have a transcription factor, in this case GlyZ, that regulates expression of all of these genes. So Nancy Keller and Antonia Rokas have identified 33 different secondary metabolite gene clusters in Aspergillus fumigatus, and we found that SLIT-A governed the expression of 16 of them, and the heat map is shown right here. So some of these gene clusters were downregulated, such as in the fumigaclavin gene cluster, and other ones were actually upregulated in the SLIT-A mutant fusarazine C, is an example. So it had been known previously by work from Nancy Keller's lab that many secondary metabolite gene clusters are regulated by the methyltransferase LAEA. However, we think that SLIT-A governs secondary metabolite genes independently of LAEA, and let me tell you why. So first off is I have put asterisks by the gene clusters that are known to be governed by LAEA, and they are shown here. So LAEA only governs 6 of the gene clusters that are regulated by SLIT-A. Also, in the LAA mutant, all gene clusters that are regulated by LAEA are downregulated, where of course some of the gene clusters that are regulated by SLIT-A are actually upregulated when SLIT-A is deleted. And furthermore, the deletion of LAEA results in a pleiotropic effect on Aspergillus fumigatus. For example, conidiation is abnormal, whereas conidiation of the SLIT-A mutant was normal. So, but to get a better look at the relationship between SLIT-A and LAEA, we looked at their relative gene expression by real-time PCR. So shown on the left-hand panel is the expression of LAEA in the wild-type strain in the SLIT-A mutant, and they are not significantly different. And the right-hand panel shows the expression of SLIT-A in AF293 and the LAA mutant, and again, they're similar. So this suggests that LAA are expressed independently of each other, and that SLIT-A and LAA govern secondary metabolite production in parallel yet independent pathways. So it's known that there is substantial heterogeneity in Aspergillus fumigatus strains. In other words, there's a lot of strain-to-strain variability in the production of secondary metabolites. So we wanted to know whether SLIT-A governed secondary metabolite in a strain of Aspergillus that was different from AF293. So Mike Bromley and his colleagues had made a transcription factor deletion mutant library in A. fumigatus A1160. And this is a derivative of strain CEA10, which is quite different from AF293. So he had sent us this deletion mutant collection. So we retrieved the SLIT-A mutant and grew the organism under low zinc and low nitrogen, and then used real-time PCR to look at the expression of secondary metabolite genes. And the heat map of the results are shown here. So on the left is the expression of genes of the SLIT-A mutant in the AF293 background, and on the right is the expression of genes of the SLIT-A mutant in the A1160 background. And as you can see, the gene expression profiles were quite similar for most of the genes that we looked at. Now, there's a few ones that were different. For example, these two genes here were differentially expressed in the SLIT-A mutant in AF293, whereas they basically were not expressed in A1160 under our in vitro conditions. This gene here was downregulated in the AF293 mutant, but actually upregulated in the A1160 mutant. Conversely, this gene here was upregulated AF293 when SLIT-A was deleted, but downregulated in A1160 in the same mutant. So this suggests that SLIT-A governs secondary metabolites in at least two different strains of A. fumigatus, but it does so at least somewhat differently. So the next question we asked was, what secondary metabolites that were regulated by SLIT-A were responsible for the virulence defect of the SLIT-A mutant? So the first one gene cluster we looked at was the fumigaclavin gene cluster. So this is known to be responsible for producing ergot alkaloids. And DMAW is a key enzyme that's very early in the pathway, and it's been found previously that deletion of DMAW blocks production of all ergot alkaloids by Aspergillus fumigatus. So we deleted DMAW and tested its virulence in steroid-treated mice. However, as you can see, mice infected with this mutant had the same virulence as the wild-type strain, indicating that DMAW is dispensable for virulence in this model. So the next gene cluster we looked at is the fumigillin-serotin supercluster. So this cluster encodes for genes that produce both of these mycotoxins. And it's regulated by the transcription factor FUMAR, which is present in this cluster. So we next deleted FUMAR and tested the virulence of this mutant. As you can see, mice infected with the FUMAR mutant also had wild-type virulence. Then we actually deleted DMAW in the FUMAR mutant, and again, found that we had wild-type virulence. So we went back and looked at our transcriptional profiling data and noticed that ASPF1 mRNA levels were quite low in the SLIT-A mutant. And so ASPF1 encodes a ribotoxin that we have previously shown is required for maximal virulence in Aspergillus fumigatus. So to see if reduced expression of ASPF1 was required was contributing to the virulence defect of the SLIT-A mutant, we replaced the promoter for ASPF1 with the GPDA promoter such that it was strongly expressed. And these are real-time PCR data demonstrating this. So here's expression of ASPF1 in the wild-type strain. This is expression in the SLIT-A mutant. You can see it's significantly reduced. And then, of course, in our overexpression strain, ASPF1 mRNA levels rose significantly. And so when we tested these strains in our assay, we found out that mice infected with the SLIT-A mutant that overexpressed ASPF1 has significantly greater virulence relative to mice infected with the SLIT-A mutant. So these results suggest that reduced expression of ASPF1 is at least one of the reasons for the virulence defect of the SLIT-A mutant. However, notice that the SLIT-A governs other secondary metabolite genes, so it's certainly possible that other mycotoxins also contribute to the virulence defect of the SLIT-A mutant. So in conclusion, nanostring technology is an efficient method for profiling Aspergillus gene expression during invasive infection. Growth in Aspergillus medium containing low zinc and low nitrogen induces a transcriptional response that is similar to invasive growth in vivo. And based on the nanostring results, we were able to identify two transcription factors with distinct roles in virulence. One is RLMA, which is critical for proliferation in the mouse one, and the other is SLID-A, which governs mycotoxin production and is required for pathogenesis. So before closing, I would like to acknowledge all the people that actually did the work. The bulk of the animal work and all the mutant construction was done by Hong Liu in my lab, but this work would not have been possible without our collaborators, especially Aaron Mitchell and his group and Vinnie Bruno and his group. So with that, I'd like to thank you for listening to my talk.
Great. Thanks very much, Scott. It's my pleasure to introduce our next MycoTalks speaker, Tihana Bicanic, who is an exemplary, internationally recognized physician scientist at St. George's University of London Institute for Infection and Immunity. Tihana has a broad research portfolio spanning the evolution of antifungal resistance in Candida and Cryptococcus, all the way to fungal genomics and host immunogenetics, and studies on Aspergillus, complicating flu and coronavirus infections. Her work is exciting and foundational and of both fundamental and clinical importance. And her outreach and work in clinical trials in Africa have been highly impactful in the context of reducing mortality of fungal infections in the HIV/AIDS pandemic. She and her colleagues recently identified alleles of the gene encoding macrophage colony stimulating factor, MCSF, that confer susceptibility to cryptococcus infection in HIV-infected individuals in South Africa. Tihana recently received the Emerging Leaders Prize from the Medical Research Foundation, and that supports studies on antifungal resistance, evolution, and mechanisms. So it's a great pleasure to welcome Tihana for her MycoTalk that's entitled Emergence and Mechanism of Fluconazole Resistance in Human Cryptococcosis.
Thank you very much, Joe, for that lovely introduction. Can you all see my screen okay? I hope so. So
you're on the split screen with the next slide up too.
Okay, is that better?
Yes, that's perfect. Thank you.
Okay, good. Right, so thank you very much. It's an absolute privilege as well as rather daunting to be part of this exciting series of MycoTalks. And as Joe has already said, I'm going to be discussing fluconazole resistance in cryptococcal meningitis, which is an opportunistic pathogen which is inhaled like many fungal pathogen and undergoes latency within the lung. But in the setting of immunosuppression, such as advanced HIV, it disseminates to the brain, causing meningitis. And it's one of the big fungal global killers, particularly in the setting of advanced HIV. And as a result, it causes an estimated 180,000 deaths every year, most of which are in sub-Saharan Africa, where Cryptococcus is also the most common cause of meningitis in HIV-positive patients. And sadly, the incidence of this disease is not decreasing despite access to treatment for HIV throughout the continent. The treatment of cryptococcal meningitis in HIV, defined through many landmark studies, initially from the mycosis study group in the United States, and then subsequently by investigators in Africa, including our own group, consists of 3 phases. An initial induction phase, which aims to sterilize the CSF using either the intravenous fungicidal polyene amphotericin B, combined with either flucytosine, the gold standard regimen, or fluconazole, or as an alternative recently demonstrated to be non-inferior in our group's ACTA trial, the combination of oral fluconazole with flucytosine. This regimen is followed by a consolidation period at moderate doses of fluconazole, during which it is aimed to start the patient on antiretroviral therapy against HIV at around 4 to 6 weeks, and then continue the patient on maintenance fluconazole until such time that the immune system is reconstituted. And this is the recommended treatment schedule in WHO and IDSA guidelines. However, in practice in Africa, amphotericin B, shown on the left, given intravenously, is not widely available, and neither is flucytosine. So fluconazole monotherapy is the norm. And fluconazole is not just used to treat cryptococcal infections, but also for oral candidiasis in HIV patients. So there's a lot of drug exposure. There are many advantages to this drug, of course. It's safe, it's cheap. It used to be donated through the Pfizer donation program. It's now available generically. It's a tablet and it penetrates very well to the site of infection. However, what happens in practice is that from the time of diagnosis, the patient is effectively on a prolonged up to 1 year course of fluconazole monotherapy, albeit initially at higher doses, then at the moderate doses during the consolidation, and then the prolonged maintenance period with low doses of fluconazole until the CD4 count is up and the viral load for HIV is suppressed. And as a result, from numerous published African series, including from our own group, the outcomes, even with higher dose fluconazole, are very poor, with very high mortality at both 10 weeks and 1 year. Now, in one of the first studies that I did on embarking on my 2-year research period in Cape Town between 2004 and 6, working with Graham Menkes and colleagues, we were able to show that in human cryptococcal meningitis, using serial lumbar punctures as shown here, done by myself on days 1, 3, 7, and 14 of treatment, the first one being pre-treatment with the drug and then on drug. And you can see that these are individual patients' rate of clearance curves, which can be summarized in a statistic called early fungicidal activity, the mean rate of clearance. And you can see just by eyeballing this, that amphotericin B is a fungicidal drug that sterilizes the CSF, whereas fluconazole at 400 milligrams a day is static. Moreover, as shown by Nikki Longley subsequently in Uganda, even higher doses at 8 or even 1200 milligrams a day, although they have some greater killing activity, they still fail to sterilize the CSF at 14 days. But why should this be a problem? Because after all, from multiple global surveys being conducted at the time using shipped isolates, published by, this is an example published by Mike Fowler and colleagues from the Artemis surveys, it seemed to be that resistance wasn't a major problem. The mode MIC, you can see here, is 4 across multiple continents and regions, and South Africa did take part in this survey. And you can see here a very low percentage of isolates with MICs above the epidemiological cutoff value of 8. Moreover, no studies have been able to demonstrate a consistent relationship between the baseline isolate fluconazole MIC and outcome in the shape of CSF sterilization, albeit most of these were conducted in the context of amphotericin B-based induction therapy, where fluconazole was just the partner drug. However, on arrival to South Africa, Graham Menkes and I noted a large number of patients presenting with relapse, clinical relapse, with secondary resistance to fluconazole developing. And this was a study that we published in CID showing that of 27 patients, the vast majority of these patients relapsing had isolates above this ECV of 8, and many were highly resistant. And indeed, since then, Felix von Gomen and colleagues have published a systematic review that includes our own cohort, comprising more than 4,000 isolates, and this is now more series from Africa, not just global series, which does seem to suggest a slightly higher prevalence of isolates with an MIC above the ECV for fluconazole. So just to summarize so far, then we can see that the majority of global cryptococcal meningitis cases occur in sub-Saharan Africa, where oral fluconazole is the most widely available treatment. That studies of drug early fungicidal activity show that the drug is fungistatic at lower doses. There seems to be little primary resistance in global surveys and no apparent relationship between MIC and clearance, and yet relapse does occur with secondary resistance in the setting of fluconazole monotherapy. So what is going on here? So as previous excellent MycoTalks have already alluded to, human fungal pathogens exhibit genomic plasticity, including aneuploidy, that is, they vary either copy number variations in regions of the chromosomes or whole chromosomes, which essentially is a generic mechanism of adaptation to stress, whereby aneuploidy provides a transient phenotypic variation, a rapid solution for coping with the stress by generating colonies that have selective advantages, a type of evolutionary bet hedging. And azole stress is just one example of this, but is highly relevant when it comes to drug resistance. And this was first described in Candida glabrata in 1992, whereby it was shown in a clinical isolate there was duplication of chromosome E harboring the ERG11 gene encoding the drug target, which was transient, that is, it disappeared on passage. Moreover, Anna Selmecki, Anya Forsch, and Judy Berman and colleagues in their seminal science paper demonstrated that in Candida albicans, the formation of an isochromosome in the left arm of chromosome 5, housing ERG11, as well as the transcriptional regulator of the Eflux pump CDR1, were responsible for azole resistance in clinical Candida albicans. Heteroresistance, which is a related concept, was first demonstrated, and this was known already in bacteriology for many decades, but Jun Kwon Chung's lab at NIH first described this phenomenon in Cryptococcus neoformans and subsequently Gattii from 1999 onwards. And this is a phenomenon whereby a minor subpopulation, shown in this photograph, is able to grow at drug levels above the MIC. Here it is at 32 milligrams per liter. And when tested, these colonies are actually resistant in an otherwise susceptible population. June went on to show that this is an intrinsic property of both neoformans and Gattii, regardless of prior exposure to fluconazole. And moreover, using CGH, she was able to demonstrate that heteroresistant populations are often disomic for various chromosomes, notably chromosome 1, which in Cryptococcus is the site of both ERG11 encoding the drug target, as well as the AFR1 Eflux ABC type transporter. So you can see here that Cryptococcus has 14 chromosomes, and this is showing in a Y-map format, where it shows copy number relative to the reference genomes for each of the 14 chromosomes. She was also able to demonstrate that on in vitro passage in higher concentrations of fluconazole yielded further aneuploidies in other chromosomes, which again were lost on in vitro passage. And this is unlike point mutations, which are stable resistance mechanisms. Ed Sionov, together with Jun Kwon Chung, then went to demonstrate that this also occurred in vivo in a murine model, whereby serially plated homogenates from mice brain, you can see that the proportion of resistant cells shown in the bottom graph as growing on fluconazole at 8 milligrams per liter developed over time, and by day 30 of the experiment formed the majority of the population. Now, heteroresistance can be measured in various ways by different investigators. So Jun Kwon Chung's group used categoric measures such as the level of heteroresistance to fluconazole, which indicates the concentration of drug, the dilution at which colonies are first seen to appear that are resistant. Or one can also use the percentage of heteroresistance by looking at the ratio of colonies growing on drug to non-drug. However, Judy Berman's lab have developed a more continuous measure, which is a reliable and reproducible in the laboratory, known as population analysis profiling. And this is shown here on the right for Candida glabrata. This is a solid drug agar method of twofold dilutions on drug and then counting colonies and then looking at the area under the curve of colonies able to grow on plates containing drug beyond the isolate's MIC. And this is known as the AUC of heteroresistance. And Judy Berman, using this technique for clinical Candida glabrata isolates, was able to show that this area under the curve correlated with efflux pump activity and expression of relevant genes in ABC transporters. Moreover, in a murine model, the highly heteroresistant isolates were more likely to persist in terms of organ fungal burden. So to summarize this section of my talk, so far what we know is that heteroresistance to fluconazole appears to be intrinsic in Cryptococcus neoformans and gattii, and this is a resistance phenotype that is not reflected in the isolate's MIC. Moreover, this is a transient phenotype. The subpopulation is selected for during therapy of in vivo infection, but seems to disappear in the absence of drug. And its mechanism, although not fully elucidated, seems to, at least in part, be explained by aneuploidy, particularly in chromosome 1, at least in the serotype A reference strain H99. So what we set out to do in my group was to look at the significance and the mechanism of this in human infection. And the work I'm now going to present is the PhD work of Dr. Neil Stone, shown here in Tanzania, where he spent 18 months of his PhD undertaking a clinical study in Dar es Salaam. And you can see here Neil undertaking the lumbar punctures, and you can also see the conditions under which this study was performed, the setting in which the lumbar punctures were performed. So this was an incredible opportunity that Neil took to undertake serial lumbar punctures in a similar fashion to what I did in Cape Town, but also saving both CSF and blood samples to measure drug levels, as well as plating out and measuring heteroresistance and serial MICs in the colonies. And he managed to enroll 2 cohorts of patients. A small cohort, 15 patients treated with standard of care fluconazole monotherapy, and then 5 patients who were part of our group's ACTA trial, who were on combination therapy with fluconazole and flucytosine. And he followed the patients up for 1 year. And this is what Neil did. At each lumbar puncture, he would take fresh CSF, take it to the lab himself, and plate it onto either YPD or YPD with escalating concentrations of fluconazole to find that level of heteroresistance and determine the percentage of heteroresistance to fluconazole. He then, for each patient, plotted curves of both the total population over time, as we had used in our trials before, but also uniquely, he also plotted, you can see here in the dotted line, the resistant subpopulation over time. He also saved the colonies growing on each of these plates for subsequent whole genome sequencing. And you can see here that this is the baseline, this is the patients. So although it looks like there are 24, there are 20 patients because some were excluded for various reasons. But there are 20 patients, and you can see here that the baseline isolate MIC, again, looks rather sensitive, looks quite uniform. There are a few with intermediate MICs, and 3 patients relapsed at 1 year, all of whom were within the fluconazole group. And this is a summary of the results of the fluconazole monotherapy cohort versus the combination therapy cohort. And again, in this diagram, the total population is shown in black, and the broken line in red shows the resistant subpopulation. And you can see here that although the total population goes down with both treatments towards day 14, when you're using fluconazole monotherapy, the heteroresistant subpopulation starts off at around 1 in 1,000, which is a standard frequency, but converges with the total population such that by day 14, it forms the majority of the fungal population in the patient's CSF. Conversely, in the combination therapy arm, this same population present at baseline at around 1 in 1,000 is suppressed by day 14. So to further characterize this, Neil then went on Neil Gow's excellent suggestion, to visit Judy Berman for a period in her lab and undertook these same population analysis profile assays using these clinical cryptococcal strains. And again, he was able to show that these isolates had a continuously distributed phenotype in terms of this light green shading shown here, which is the area under the curve of heteroresistance. And you can see here, there are also the darker and the bright green, which are the susceptible and tolerant populations. And I won't go into these at the moment, but if you want to know more about this, do listen to Judy Berman's excellent MycoTalk on this topic. So Neil undertook these measurements for all of the 20 isolates, and he then correlated this area under the curve of heteroresistance with a phenotypic assay of efflux pump activity known as a rhodamine 6G assay. And I'm not sure how clearly you can see, I hope that you can, but this is a red dye that in the presence of glucose is pumped out of the cell using efflux pumps. And on the left, you can see this end of the experiment for a highly heteroresistant isolate that's disomic for chromosome 1. And you can see there aren't many cells left containing red dye. And on the right, you can see an isolate that's euploid and has a lower level of heteroresistance. And in this case, there is more dye retained within the cells. And indeed, when he plotted the AUC of heteroresistance against the RCG efflux normalized to H99, there's a moderate but significant correlation showing that essentially this area under the curve does correlate with this efflux pump activity phenotype. Moreover, when he undertook RT-PCR looking for gene expression of AFR1 and ERG11, shown here on the bottom, he was able to show that relative to H99 and relative to the euploid isolate, the highly heteroresistant disomic isolate showed an approximately 2-fold increase in expression of these relevant genes. He then went on to undertake whole genome sequences. And just to show you again that Neil saved colonies from both the unselected YPD plate as well as the drug plate, the fluconazole or heteroresistant colonies. And all of these at baseline before any treatment were subjected to whole genome sequences. And what I'm showing you here is for those 20 patients, what I'm showing you here is the unselected colonies Y maps on the left. And you can see that in the unselected colonies, there is aneuploidy in just 1 out of the 20 isolates of chromosome 1. But look at that heteroresistant subpopulation, and just remember, this is before any drug treatment. So you can see here that before exposure to fluconazole, there are a lot of isolates with aneuploidy at baseline. So this is intrinsic, and it's not just chromosome 1. You can see there on the right, on the top, a number of different chromosomes. Indeed, when you look at the 20 heteroresistant isolates, 12 of those were aneuploid and 8 did not have aneuploidy. And of those 12 with aneuploidy, 9 were aneuploidy of chromosome 1. And he then went on to also follow the patients for a year. And in those, this is one of the 3 patients who relapsed. And you can see here that at relapse, the MIC went up over time. So it's shown at 3 months and 1 year, 2 episodes of culture positive relapse. And also corresponding to this, there is serial acquisition of disomies, initially of chromosome 1, and these are unselected colonies, not growing on fluconazole. These are non-drug, and there is aneuploidy of chromosome 1 and 12 at day 90, and 1, 4, and 12 at 1 year, corresponding to a resistant MIC, so an emergence of resistance. He was also able to show on passaging these isolates in Judy Berman's lab in liquid culture over 10 to 12 days that this isolate then reverted to the original phenotype without any disomies, and indeed reverted to sensitivity to fluconazole, as shown here on E-test. So this would suggest that this aneuploidian resistance might carry a fitness cost. However, these isolates clearly were still able to cause disease and cause clinical relapse in this patient. And we also went on to further assess whether there was any fitness cost for these isolates through growth in YPD agar and robotic phenotyping at Duke in John Perfect's lab. And in this assay, we were not able to find any differences in vitro growth rates between the unselected and the heteroresistant colonies, regardless of the presence of aneuploidy. We then tried to do a preliminary characterization, although this definitely requires further work, of the 8 isolates with apparent non-aneuploid mechanisms of heteroresistance. And to do this, we looked for, together with Joe Rhodes from Matt Fisher's group, for non-synonymous SNPs that were unique to the heteroresistant colonies, as well as seen in any post-treatment isolates, because Neil did save any post-treatment isolates. And you can see them here. Most of these are genes in seemingly unrelated biochemical pathways, and we are uncertain of the functional significance of this. And the one we thought was potentially the most interesting was the RAD57 SNPs, because RAD57 is a DNA repair protein, and this would certainly need further validation to see whether this indeed plays a role. However, given only a subpopulation is resistant and the transience of this phenomenon, it is less likely that fixed mutations are playing a role here. And perhaps what's crucial here is some sort of regulation of gene expression. And I just want to also emphasize that we did look for SNPs in mismatch repair genes, MSH2, and we also looked for SNPs in apoptosis inducing factor, AIF, and did not find these in these 8 isolates. So we have established, and this is one of the fantastic things about this isolate collection that were collected in this unique way, is that we are able to share this resource linked to clinical data with the mycology community. And we have sent these isolates to Liz Ballou, who's now at Exeter, who was at Birmingham, who has been undertaking her Titanization assay, more of which you can hear about in her MycoTalk, with the hypothesis that Titan cells, which are polyploid and are able to give rise to aneuploid progeny, that the ability to titanize may be related to becoming hetero-resistant. Robin Allshire, who's a professor of chromosome biology and expert on heterochromatin and epigenetics, has also got the isolates to look at whether formation of heterochromatin over resistance loci resulting in their repression could be responsible and in some way influence evolution of resistance. And lastly, Joe Heitman is going to undertake a small RNA sequencing and a series of other experiments, including genetic crosses and animal models, to look at whether perhaps this unstable resistance may be due to formation of small interfering RNAs against a relevant gene. And if you want to know more about the work they've done to elucidate this mechanism in Mucor against FKS, watch Joe's excellent MycoTalk. Now, of course, resistance and response to the drug is not just about whether an isolate is susceptible or resistant. You also need adequate drug exposure. And this is measured in pharmacokinetic, pharmacodynamic studies. So this is a study done with William Hope's lab in collaboration. He was my co-supervisor for Neil. And this is some of William's prior published work, bridging murine work to determine a fluconazole pharmacodynamic target in humans. And the pharmacodynamic target for azoles is the area under the concentration curve over the MIC. And this graph is here just to show you that this solid line here, at a dose of 1200 milligrams a day using isolates of ranging MICs in mice, when bridged to humans, you can see here in these arrows that an MIC, an adequate pharmacodynamic target would be attained of isolates with an MIC of 2 in just under 2/3 of patients, whereas isolates with an MIC of 4 or above, less than 1%, would actually attain this target. And we went on to show this William went on to show this in a murine model, whereby mice were infected with H99 and then treated with fluconazole. You can see the controls on the left and the fluconazole treated on the right. And on the left, you can see the total population, again, solid line. Dashed line is the resistant subpopulation, which in untreated mice continues in a similar ratio to the total population, about 1 in 1,000, 1 in 10,000. However, the two curves converge, just like in the human, in the mice. And moreover, on sequencing of these colonies, again, disomy is observed in brain homogenase, disomy of chromosome 1. William then went on to perform something known as Monte Carlo simulations, which is using data on drug levels in the CSF and plasma from 11 of Neil's patients, married to the rate of clearance of infection for these same patients. And he then projected these data to a hypothetical, much larger population of 10,000 individuals treated with fluconazole, based on what we know are the pharmacodynamic targets. And again, you can see here on the right, the total fungal burden and the resistant subpopulation for this hypothetical population treated over 2 weeks with fluconazole 1200. And this shows the centiles. And after 2 weeks, this population, only 13% sterilized their CSF with this fluconazole dose, and 83% had resistant colonies detectable. So in summary, fluconazole heteroresistance is an intrinsic property of clinical cryptococcal strains that is not captured using conventional methods and amplified under drug pressure. Aneuploidy appears to be the predominant, but not only mechanism now demonstrated both in mice and in human infection. Other mechanisms, particularly epigenetic, need further exploration. Resistance emergence cannot be prevented using clinically relevant doses and explains the fungistatic pattern of fluconazole for cryptococcosis. The use of combinations therapy, in our case, fluconazole and flucytosine, can prevent this. And I think this draws our attention to a much larger issue in the sense that, as we all know, azole use is ubiquitous for fungi with emerging resistance. And it is very important to develop and deploy both novel and existing drugs in a way that prevents resistance emergence, with a role for combination therapy. And just lastly, to acknowledge everyone involved, including Neil Stone, and this is him shown getting his PhD, Judy Berman and William Hope, who co-supervised him, my African collaborators, Graham Menkes and Sayokim Finanga in Tanzania, June Kwon-chung, and also Tom Harrison at George's, my longtime mentor, without whom any of this would not have been possible, and the funders. Thank you very much.
Fantastic. Thanks so much, Tihana. So I think Iliyan will lead off the questions, starting with questions for Scott, and then we will trade back and forth.
Right. Sorry. Okay. Okay, I can start it now. Question for Scott. So there is this down regulation, very strong regulation in the DHN melanin cluster that is happening in the SLEET mutants. So questions from Gordon Brown and George Mick, is that actually linked to virulence? Have you explored that direction?
Yes, we noticed in our transcriptional profiling data that the DHN melanin cluster was downregulated. But surprisingly enough, when we grew our organisms on plates, we did not see white conidia. The conidia were normal colored. And as you know, the melanin pathway is particularly important for resistance of the conidia to being killed by phagocytes. And as I showed you, the macrophage killing was normal. So I don't really know what to say about this downregulation. We definitely saw it, but we don't know the significance of it. And I don't know if it contributes to reduced virulence.
I see. And I will go with a second question on the downregulation, and then I will pass to Joe. Very related question to downregulation, but this time in the ergosterol cluster. Is there a link with viral virulence, and have you tried to regulate the ergosterol levels to see what that means?
Yeah, we just found this paper that was that described the reduced ergosterol pathway expression in the SLEET mutant was published only a month ago. So it's an intriguing possibility, but we haven't investigated it yet.
Thank you, Scott.
Great. Tihana, we have a question about the formation of the aneuploidies, and that these can arise in response to exposure to different host niches. And so is it possible to distinguish if the heteroresistance is arising from exposure to the host or exposure to the fluconazole?
I think that's an extremely important question and a very valid one. And certainly it's something we thought a lot about, both when analyzing the murine experiments and the human data. I mean, it's also one of the other worries is, is plating out on fluconazole itself inducing the formation of aneuploidies, and how do you get around that? Because we know that the host environment induces aneuploidy. We also know that the drug itself alters cell cycle progression and has been shown in Candida to result in trimeras that can result in tetraploids and aneuploids. And similarly in cryptococcus, that maybe fluconazole can induce titanization. However, I think the fact that, and certainly in the mouse, I skirted over it, but if you look closely at the slide with where we plated out the murine brains it was plated out on fluconazole, but even the untreated mice did have some aneuploidy of chromosome 1, albeit when grown on fluconazole-containing plates. So I think this is quite difficult to tease out. I mean, I think minimizing passages, looking at which aneuploidies disappear in the absence of drug are important ones, looking at plausible mechanisms in terms of certainly chromosome 1 and drug targets. But I completely agree that aneuploidy, clearly even in the environmental isolates of Cryptococcus also show aneuploidy. So clearly there are a number of different stresses, both in the environment and in the host, that drive aneuploidies. And I would just say this is a generic mechanism, and all we've been able to show is that fluconazole potentiates that, and combination therapy appears to suppress it.
So that's a good lead in to the next question, which is from Gordon Brown. Can the resistance arise through similar mechanisms when you're treating patients with the drug combinations?
Well, we haven't, I mean, we looked at it obviously in this very small cohort of 5 patients with ACTA. Certainly in the other combination studies we have done in South Africa and subsequently in other parts of Africa, using oral therapy and amphotericin B-based therapy and drug regimens that are associated with rapid clearance, we've seen far less relapse, and we've seen much less secondary resistance. However, resistance can arise. So Neil in the laboratory with working with Judy Berman tried to look at some time kill assays using drugs alone or in combination to try and replicate what was going on in the patients. And he did find that he was able to replicate the emergence of heteroresistance. And when using flucytosine for one isolate, he was able to suppress it, but another isolate, possibly related to fluconazole exposure, then went on to develop flucytosine resistance. So I guess, would the mechanisms be the same? No, it depends on the drug and its resistance mechanism. So certainly it is possible that other mutation-based resistance could emerge using combination therapies to the second drug.
Okay, thank you very much. Over to you, Iliyan. Iliyan, you're muted.
All right. So Scott, there is a lot of interest in the nanostring technology and the application of it. So I combined a few questions here from a few people. So can you possibly use the nanostring technology to distinguish commensal versus invasive stage with, and then kind of use that to stratify by basically patients that will be treated? A related question to that, can you adapt the nanostring technology basically for a dual use? So looking what happens at the Aspergillus and at the host in the same time.
So obviously looking at gene expression profile of Aspergillus to determine whether it's a commensal or invasive organism would be really helpful. Probably the issue is that these are mouse studies where we grind up the entire lung, and that would obviously not be possible for humans. So that would probably be the biggest issue. But the question, I guess, is if you recovered organisms from BAL, could you use it? Maybe, but typically though, putting on my clinician hat, you get very, very few organisms by BAL. Typically when you grow in a human, if you played out the BAL, you might get only 1 or 2 colonies of Aspergillus. So it'd be pretty hard to do nanostring on that because it'd be growing in vitro for a while. So probably not so useful. But you can easily use nanostring to measure the host mRNA response, although you only get a snapshot, right? The limitation of nanostring technology is we were able to measure 400 genes or so, 474. And so it might be better to do a dual approach where you extract the RNA and use nanostring to measure your fungal gene expression, but use RNA-seq to measure the host gene expression. And also, a nanostring has actually been adapted for using single-cell transcriptomics I've never done it myself, but in theory, you could probably measure not only the transcription profile of a single cell, but maybe the fungus within that cell as well.
Excellent. So related to technology, so now we are moving to the meta-analysis. So one question here, actually a couple of questions. So for the meta-analysis of transcriptomes, how did you treat the data to allow the cross-platform comparison if you have to, compare the microarray versus non-string data? And another related question, so did you try to validate actually the expression data in SLE mutant with mass spec?
Yeah, so for the first one, we just did gene expression ratios. And so, not a perfect way to go, but probably the best way you can do it. And no, we didn't do mass spec to look at the decrease in secondary metabolite production. But let me say, I think that that's probably the reason why the organism had a reduced capacity to damage epithelial cells, is by reduced production of secondary metabolites. So we found, for example, the LEA mutant also has reduced capacity to damage pulmonary epithelial cells.
Thank you, Scott. So I'm handing to Joe now.
Okay, Tihana, we have a question from Feng Yang about the aneuploid hetero-resistant strains and whether you've tested them for susceptibility to other drugs.
The short answer, and Neil, I think should be listening, but I don't think that we have. What we have done, like I said, is we've looked at combination of second drugs with fluconazole in vitro, and the ability for different, what Judy calls adjuvant drugs, to tackle these heteroresistant colonies. And interestingly, and this is also relevant to Joe's work on Mucor, but the combination with FKS 506, and Neil looked at various cell signaling pathway inhibitors, and he did find some effect of FKS 506 and calcium-calcineurin pathway inhibitors were good adjuvant drugs with fluconazole against these isolates, obviously in addition to flucytosine. But if your question, Feng, was about did we test them for other azole susceptibility, we did not, although we have since profiled some of these isolates for the new tetrazole antifungal Viamet, and they seem to be hetero-resistant to this drug as well. This is unpublished data, but otherwise we've not looked at MIC to any other drugs, so I'd welcome any suggestions.
Great. We have another question about the possibility that there could be host factors that are involved in the heteroresistance process. And are there any correlations for example, for immune factors that might be contributing to the emergence?
Yes, I saw that, Peter. Thank you for that question and your comment. I think that's a really good question. And interestingly, as I was rereading Neil's thesis in preparation for this talk, I noticed Neil looked at trying to correlate. Obviously, it's a very small cohort. We haven't as yet, but we have the CSF in the freezer, including the cell pellets of the fungi, by the way, but we haven't measured the CSF immune responses in this patient cohort. I mean, we know from our other work that interferon gamma is very important in host clearance. But interestingly, Neil looked at things like fungal burden, altered mental status, mortality, didn't find any correlations. But the one thing that did correlate with percentage heteroresistance was receipt of antiretroviral therapy. So, and I was WhatsApping Neil just this morning, and we were discussing why do we think they had more heteroresistant, and maybe it is a host immune effect, or perhaps those patients, unbeknownst to us, were more likely to have been exposed to fluconazole. But there might be some immune drivers, and maybe we should look at the CSF cytokines from this population to tease this out further, or anything else you might suggest, Peter.
Great. Back over to you, Iliyan.
All right. So I'm combining here questions from Rob Kramer and from Nita Schlitzinger. So it's about like the early time points of infections, basically. So EF293, does it invade the lung parenchyma in the cortisone acetate model at days 4 or 5? And does it form biofilms with leukocyte infiltrates in the large airways?
Yeah, it actually invades quite a bit. So at our time, by our histopathology, you can see that it's deeply invaded, forming what looks almost like microcolonies in the lung with a central area with hyphae going out from them. So yes, there's deep invasion for sure.
And related questions. So what condition is hypoxia actually in your model?
Yeah, so we don't have the high-tech hypoxia chamber that Rob Kramer has, but we bought the hypoxia packs for doing bacterial anaerobe work and use that to deplete the oxygen from our cultures.
All right. So I will hand to Joe now.
So, Tihana, I was very interested in the study of the whole genome sequences and the mutations that were identified in the non-aneuploid isolates. And I'm curious if you may have done the same type of analysis for the aneuploids. And what I'm curious about, Angelica Ammon's lab in Saccharomyces had found that you can identify mutations that let strains tolerate aneuploidy to a greater extent. And I'm wondering if that might be occurring either because of natural variation across isolates, or if there's enough selective pressure to maintain the aneuploid chromosome that you're also getting SNPs that support that.
No, again, Neil can confirm this, but the data we presented was in the paper was from the non-aneuploid isolates, and that would be something that would be really good to look at because we do have those sequences available. I'm not sure whether Joe Rhodes did look at that whole population, although actually thinking about it, the table from our paper, Neil, do you want to comment as well in the chat? We may have looked at it also for the aneuploid colonies. Let me bring the paper up just now. Yes, no, we did look for them, actually. We looked for aneuploid heteroresistant colonies and the euploid, and we looked for mutations, non-synonymous SNPs in the specific to the heteroresistant colonies. But I'm sure there are other genes that we should be looking for and could in collaboration with you, Joe.
So one of the things that Angelica's lab had found was mutations that alter activity of the proteasome. And the thought was maybe you were activating proteolysis of proteins that are overexpressed by the aneuploids. I want to return to the question about Titan cells and Titanization and your discussions with Liz Ballou. If I remember right, there are studies maybe from Kirsten Nielsen's lab where they found when Titan cells produce progeny, a lot of those progeny can be aneuploid. And what I don't remember is what chromosomes they were aneuploid for and whether there might be any pattern from the Titan cells producing aneuploids in vitro and the aneuploids you see from the patients in Africa that might give further weight to the idea that a Titan cell might be an intermediate in that process.
Yeah, I mean, I think this is what, in fact, I had an email correspondence with Liz again in preparing this talk, and I think she may still be on this MycoTalk, but essentially what she's been trying to look at, I'm not sure, to be honest, I don't know which chromosomes the Titan cells are aneuploid for. I don't know that. I do know, maybe she can comment on that. I do know that Titan cells have been known to form, and Kirsten Nielsen has shown in response to fluconazole. But what so far in the preliminary data that that Liz has shared with us and presented as a poster so far is of the clinical isolates that have been analyzed, some isolates were able to titanize and others aren't. And of the fluconazole resistant ones, when they looked at them, they weren't resistant anymore, consistent with this transient phenotype. This is the challenge of studying it. And some had gained the ability to titanize, others had lost this ability, and this seemed to be very reproducible for the strains over time. So as ever, and then they are looking at various in the whole genome sequencing, they're analyzing SNPs that are associated with ability to titanize or not. And they're looking at SNPs in the trehalose synthesis pathway, and they're evaluating this further. So it's watch this space for that. But as ever with these things, it's not a straightforward black and white answer with titanization.
Terrific. All right, back over to Iliyan.
All right, so a question from Eleni Bingo. So how would you interpret beautiful data on the steady state, but how would you interpret actually findings from nanostring during infection? And related question, going more into the infection direction, basically, can you clarify the impact of SLIT-A on virulence in systemic infection in competent hosts versus infection in immunocompromised hosts? That's from Gordon Brown.
Yeah, so to answer the first one, we think the steady state means that the organism is being exposed to pretty much the same conditions at days 4 and 5. In other words, it's growing into the tissues and of course being attacked by phagocytes. And because these mice are so immunosuppressed with the steroids, it's sort of a battle between the host and the fungus is clearly winning. We picked day 5 because we know that by day 6 and 7, the mice start dying. And so this is sort of the end stage of the infection, which, as I mentioned, is likely different from what's going on at the beginning. So in terms of immunocompetent mice, mice are very, very resistant to Aspergillus, which actually makes sense. Think about it. Mice sort of run around sniffing moldy things. And if they died every time they sniffed some Aspergillus, there wouldn't be mice around. And so immunocompetent mice, you have to give them a huge amount of Aspergillus intratracheally. And even then, maybe only 5 or 10% would die. So when you have mutants that are attenuated in virulence, it's virtually impossible to study them in immunocompetent mice. So we haven't looked at this.
All right, and the second question from Ted White regarding the SET-MET clusters. Any indication that the clusters might be tissue-specific or is associated with immune reaction or something else? So cells don't usually do a lot of gene expression without reason, although I get that one cluster does seems to have an effect.
Yeah. Well, if you think about it, though, Aspergillus did not evolve to kill people, right? So we're sort of an accidental host. In fact, some elegant work by Axel Brockhage and his group has suggested that the main defenses of Aspergillus are actually to prevent it being killed by free-living amoeba that live in the compost pile. So the genes it expressed when it's engulfed by a human macrophages are probably triggered because it thinks it's inside a free-living amoeba. So those secondary metabolites may not be very helpful, or at least some of them may not be helpful for it to survive in the human host. They might help it survive when it gets engulfed by an amoeba. And so I think that the response is not as finely tuned as we would expect for something like Candida albicans, which of course has been evolving to grow within humans.
All right. Thank you, Scott. So we are approaching 1:30. If there is a very quick last question, Joe, or otherwise we'll be wrapping up.
I think we should thank our 2 esteemed physician scientist colleagues for stellar scintillating talks in the MycoTalks series. And the clarity of these presentations were just outstanding and very, very exciting work. I'm looking forward to the next chapters.
Absolutely, beautiful work, and it spanned from basic to clinical research on few continents. So it's amazing. That's what the MycoTalks are about. Thank you very much, and thanks to the audience.
Thank you.